Okay, once again, welcome back to the class. So we were discussing on industrial applications of microorganisms. So in that, we were discussing about sauerkraut. Sauerkraut is a product which is prepared from fermentation of cabbage. So now let us know the procedure of preparation of sauerkraut. So material, I think, I'm sorry, gone back. One second. So the materials, I have already told you what are all the materials required for preparation of sauerkraut. So we require cabbage, we require salt or NaCl, polythene covers and beakers. So if you have these materials, so the procedure goes like this. Take a good cabbage. So remove the outer solid layer, the outer layer which are absorbed with the soil and other things which are harder. Remove that, one or two leaves. So then you cut and remove the inner solid rind. Inside at the middle, there will be a solid rind. You remove that one and cut the cabbage into small pieces or shreds. And this shredded cabbage are mixed with the salt powder. So take a tray, put them, and by weight basis, if for every 100 gram, put 2.5 gram of salt or NaCl and mix it thoroughly. So after that, so the mixture will be transferred into a beaker. Take either a glass beaker or a plastic beaker or a jug type of material. So fill into that. After that, so you need to take a cover, polythene cover. So fill that with a water, tie it. So tie that one and place it on a beaker. So they compact the content of the beaker to press the contents of the beaker, that is shredded cabbage which is mixed with the salt. So and to seal the opening, to seal the opening of mouth of the beaker. So we are filling a plastic cover. So with the water and we are tying its mouth and putting it, placing it on the beaker containing the shredded cabbage. So this will, what it will do is, this will put a mechanical pressure on the cabbage, so which will help in the expelling the juice from the cabbage cell. So which will act as a sugar for the sugar or nutrients for the fermenting microorganisms. After this, you place this entire setup, you place in a dry, cool place for 4 to 6 weeks. That is 1 to 1.5 months. It is a long period. Place this in a cool, dry place. So with a temperature of less than 25 degrees centigrade. So for 4 to 6 weeks. And once in a week, you need to check the beaker for any rancidity or rancid smell or a pungent smell or any bad odor. So because of contamination, if there is any bad odor coming out of that, you need to check it. And after 4 to 6 weeks, so the sauerkraut will be ready. After that, you can take out. You can take out and you can put it in a glass bottle, airtight glass bottles, or even you can put it in a polythene bags. So you can store in a normal condition or even in refrigerator, you can store. So and you can use it for a consumption. So this is the procedure for preparation of sauerkraut. So you may get the questions about this in your exams. Another tempeh. In one of the exams, they have asked tempeh, although tempeh was not there in the syllabus, but they have asked. That's why I'm covering this. Tempeh is a fermented cooked soybean product prepared by using one fungal fermentation. That means here what they do is, so they cook soybean dals, dal that is prepared from soybeans or soya dals. So after cooking, so it is mixed with a fungus, one fungus. So and it is allowed to ferment. The fungus will grow and cover all the dals, all that is dals, and it will produce some characteristic taste. So that is called as a tempeh. Okay. Tempeh is a soybean food made by controlled fermentation of cooked soybean. Cook ಆಗಿರುವಂತ soybean. Controlled fermentation of cooked soybean with a rhizopus mold. With a mold called as Rhizopus oligosporus, which is also called as a tempeh starter. So this fermentation binds the soybean into a compact white cake. So here you can, the picture you can see. So the mycelia of rhizopus will grow all around, inside and outside the soybean. It will ferment and will produce a compact cake-like thing. This is called as a tempeh. And tempeh is originated from Indonesia. Indonesia. So even today, till today, this tempeh is very famous in Indonesia. And tempeh is prepared or fermented using a fungus called Rhizopus oligosporus. You need to remember this. And this is of high protein food. This contains very high protein. So this fermented food will be like a non-vegetarian. It feels like once you eat, it feels like a non-vegetarian type of food. So it will be somewhat fibery type. It will be somewhat fibery type and chewing type. You need to chew that to eat that one. So it has a chewy texture. Because of this, the vegetarians who are pure vegetarians prepare many of the products like chili chicken, so butter chicken like that, or whatever the dishes they made from chicken. So the vegetarian prepare same dishes using a tempeh. Because this is because the texture of this is similar to non-vegetarian foods. Okay. So in the United States, it is used as a meat alternative because of its chewy texture. As a result, chewy texture and it has a distinct flavor also. As a result, a wide variety of tempeh-based meat analogs are available in the United States. Tempeh can be further processed and pasteurized for distribution. Okay. So this is a tempeh. We can see here. So here the soybean will be boiled. You will know the procedure how to prepare boiled. After boiling, so it will be mixed. After boiling, it is dried by frying in a pan. So it is cooked. The water is removed. It is fried once again on the tawa or pan to remove the excess whatever the moisture is there. Then it is mixed, cooled. After cooling, it is mixed with the starter culture. Starter culture means fungal culture, Rhizopus oligosporus. It is thoroughly mixed. After that, it is tightly packed. So in a cover, it is tightly packed and it is put it in a oven. So to allow for the growth of fungi and fermentation. After few days, after 2 to 3 days, so there will be a complete growth of fungi. So it will form a complete compact cake type material or a burfi type material which can be cut and it can be cooked and used. Okay. So this is the procedure for preparation of tempeh. Soak the soybean in 2 liters of water for almost 1 day, 6 to 18 hours. You need to soak the soybean. Put the beans in a cooking pot or a utensil. Add enough water. Add vinegar. Little vinegar and cook for 30 minutes. It is boiled for 30 minutes. Soak it, boiled for 30 minutes. Drain off the water and dry the soybeans or remove the excess water by frying in a medium flow for a few minutes until the beans become dry. Allow the soybean, the fried ones, to cool below 35 degrees centigrade. Sprinkle with Rhizopus oligosporus starter culture. So then mix it uniformly. Mix uniformly using a clean spoon at least for 1 to 2 minutes. And then it is packed in a polythene cover. That is especially perforated. The polythene cover should be perforated or there should be holes for aeration. Press them, place in incubator around 30 degrees centigrade for 2 days. 36 to 48 hours, that is 2 days. So during which the tempeh fermentation takes place, then the container should be filled completely with the white mycelium and the entire contents can be lifted out as a whole piece. And about tempeh, what will happen? What are the advantages of this tempeh fermentation? It is an excellent source of protein. Contains all the. It is known to contain all the essential amino acids. And it contains the same quality protein as a poultry meat. So it can be used as an alternative to poultry meat. It is an excellent source of calcium. It is low in saturated fat, high in essential fatty acids and vitamin. It is cholesterol free, unlike meat. It is high in soluble dietary fiber is also good for sugar patients. Easy to digest. So after fermentation, the protein or fiber, whatever is present in the soybean, has become a simpler form. It is easy to digest because fermentation process break down the complex protein found in soybeans, making tempeh more easily digested than the unfermented soybean. It is high in isoflavones. Isoflavones are known to prevent the cancer. It is a good source of folic acid. It is a good source of folic acid, good for pregnant women. And it is low in sodium, so good for hypertension people also. Okay, this is about tempeh. In beverage industries, so you know that microorganisms are used in the production of alcohol. So in alcohol, what will happen? Whatever the alcohol is done using potato or barley or wheat or grapes or any other foods, so which contain sugars or carbohydrates. So here carbohydrates will be converted into alcohol and acids. Okay, there are different procedure for preparation of different types of beverages. So beer is the non-fermented beverages. The non-fermented beverages are beer and wine. So here directly the product we will put the yeast. We will put the yeast, and here carbohydrate will be converted into sugar and alcohol. So there will not be any distillation. We are not going to concentrate it to increase the alcohol percentage. So beer, there are two types of yeast are used in beer preparation. One is called as a top yeast, another one is called as a bottom yeast. Saccharomyces cerevisiae and Saccharomyces carlabergensis. Saccharomyces cerevisiae is a top yeast, which is also called A. Saccharomyces carlabergensis is a bottom yeast. And wine is produced by a specialized yeast called as Saccharomyces ellipsoides. And sake is a wine prepared from rice. It's called as a rice wine, especially produced in Japan and northeastern states. Our northeastern state, in every home they prepare this rice wine. It is called as a sake. It is prepared from Streptomyces species and Aspergillus oryzae. But here they are not going to add any microorganism. It is naturally present on the natural product. 1 second. I'll take 1 minute break.